Hello, and welcome to this Nature Research custom webcast titled Single Molecule Proteomics. My name is Jayshan Karpan, and I will be your moderator. Today's webcast is produced by GenScript Biotech. We'll begin the webcast with a presentation from Chirlmin of Delft University of Technology, and then end with a Q&A session. To ask a question, just type it in where it says type your question here and then press submit at any point during the webcast and we will answer them today. And now over to Chirlmin.
Thank you, Jason. As introduced, I'm Chirlmin from Delft University of Technology. In this webcast, I'm going to present the emerging landscape of a new research field, single molecule protein sequencing. I will start with briefly reviewing the progress in DNA sequencing technology that was made last 2 decades. Near the end of 20th century, whole genome sequencing drew a large attention. The complete human genome became available in 2003. After this achievement, which had seemed to be impossible before then, the cost of sequencing a human genome has dropped rapidly and has become below $1,000, making it affordable to use it for research and clinical applications. It is now even possible to do DNA sequencing at the single molecule level using a platform of single molecule fluorescence from Pacific Biosciences and a platform of nanopores from Oxford Nanopore Technologies. I should say that this ultimate sensitivity has enabled single-cell genomics and transcriptomics for disease screening and diagnostics. This is a remarkable achievement we have observed the last 15 years. But what about proteomics? Proteins constitute molecular components involved in all the essential biological or biochemical reactions of living cells, such as DNA replication, energy generation, and force generation. It is needless to say that protein analysis is essential for our understanding of the molecular biology of the cells. But both accurate and comprehensive sequencing of proteomics remains an outstanding challenge. Here I want to remind you of the central dogma of molecular biology. DNA is the blueprint of the cell. In case of human cell, there are about 20,000 genes, and a simple picture we had earlier was these 20,000 genes might encode for 20,000 proteins. But this one-to-one correspondence is too naive a picture, as the 20,000 genes are translated into a surprising number of 1 million different proteins due to alternative splicing and post-translational modifications. It illustrates clearly that we can understand biological processes only by looking at the genome, transcriptomes, and proteomics all together, not just one of them. Global proteomics requires a very sensitive tool. The golden standard of such analysis is mass spectrometry, for being the most sensitive technique in the market. But it has an intrinsic limitation that it measures only a single piece of information from a molecule, the mass to charge ratio. Unfortunately, it discards other rich information, such as the order of amino acid monomers. Due to this intrinsic limitation, it requires a large amount of cells for protein identification. The dynamic range of standard mass spectrometry is, well, as high as 10 to the 5th, which is a remarkable achievement from decades of work, but it is still lower than that of proteome, 10 to the 8th to 10 to the 9th. It is also a spectral analysis, so data interpretation is not straightforward. It is not an easy machine to operate and is very expensive to afford and maintain. With all these concerns, what biophysicists are dreaming of is to develop an entirely new platform, which allows for single molecule, single protein analysis, which will enable us to detect low abundant proteins such as signaling proteins. And with such a sensitive technique, we'll be able to sense and classify ultra low level of protein markers from bodily fluid. There are several different proposals on developing single molecule protein sequencing tools. In today's presentation, I will mainly cover single molecule fluorescence approaches and briefly nanopore method with one example. Other methods, such as advanced mass spectrometry, will not be discussed, but there's a review paper that I'm preparing with Javier Alfaro and Amin Merler. I want you to wait for this review paper coming out. To begin with, let's review our challenges. The immediate difficulty is how we can distinguish 20 or larger number of different amino acids. There are not 20 orthogonal chemistries that allow for distinctively labeled 20 different amino acids. Even if we can label them with different chemicals or fluorophores, current single molecule techniques do not have a capability of differentiating 20 different signals. But another way of looking at this problem is to question whether we need to do sequencing a protein at all, particularly de novo sequencing. If the sequence of a genome is known, it can be predicted what proteins will be expressed. So just like how mass spectrometry data analysis is done, what we may need to know for protein identification is a small fraction of information from a protein. This leads to the concept of single molecule protein fingerprinting. We may label only a few types of amino acids, such as C's and K's. And in case we read the order of C and K's in series, back of envelope calculation suggests that we may need only about 15 of them to identify a sequence out of 20,000 genes. We have done a computational analysis in collaboration with Dick de Ridder from Wageningen University in the Netherlands. The analysis, which was done using a human proteome database, suggests that when there's no error, 90% of proteins are precisely identified. When the errors of deletion, insertion, swapping take place, the prediction power drops down, but this can be rescued by using extra information, such as the distance between C's and K's. It's shown in the graph with a label of CK dist. This can be, it can be also extra signals from a third or fourth amino acid. To demonstrate this fingerprinting, my group decided to use a method of molecular scanning. Here we have a peptide that is labeled on its cysteine and lysine residues in two different colors. We use a molecular machinery from the nature, bacterial chaperone protein ClipXP, which pulls a protein or peptide into its central nanochannel using the ATP energy. This molecular scanner is labeled with another color of fluorophore, which allows for the use of FRET. So what is FRET? Briefly, FRET involves two fluorophores, and when a donor, which can be directly excited by an external energy source, is placed alone, it emits its own photons. When an acceptor of fluorophore of a different color is present within a few nanometers, the energy is transferred from the donor to acceptor, and it becomes the acceptor that emits photons in a different color. This allows for nanometer resolution, and this nanometer rule of FRET together with the moving scanner, enable us to determine the order of C and K in real time, as animated here. For a proof of concept, we surface immobilized ClipXP, which was labeled with the donor in green. We then introduced a synthetic peptide, which contains a single cysteine tagged with an acceptor in red. The time trace on the top shows that there are a constant green signals recorded until the peptide arrives, which we can tell from the second panel, and until the acceptor approaches the ClipP chamber, which is the bottom chamber, and the acceptor takes the energy from the donor. After that, the peptide gets cleaved by ClipP, a protease, and the fresh signal disappears. And this demonstrates that we can use this molecular scanning scheme for peptide analysis. And then we extended this to protein analysis using a model protein by titin monomer. This particular protein has two cysteine residues, and so it's labeled with the two acceptor molecules. The acceptors are separated by 16 amino acids, and as you can see from the top trace and also the third trace, there are two peaks observed sequentially. To confirm that these two peaks are from the two cysteine residues, we adjusted the ATP concentration. And in the bottom figure, you can see that the time between the two peaks is dependent on ATP concentration, indicating that this is truly a protein translocation process. This molecular scanning is one of the ways of using single molecule fluorescence for protein sequencing. And there can be several different ways. An intuitive way of using single molecule fluorescence is to directly observe the profile of a protein and the locations of amino acids. But since most proteins are coiled within a few nanometers of space, we will first have to stretch it using an external force. And there are a couple of ways of doing it, for example, by using optical or magnetic tweezers. Supposedly we managed to stretch that, it is still challenging to image the linear profile since the distance between monomers is only 0.4 nanometers, which cannot be optically resolved when a conventional light microscopy is used. But Peng-In and Ming-Jae Da from Harvard has proposed is to use nanoscopy. Nanoscopy uses the photo-blinking nature of fluorophores for single molecule localization. And then, well, the question is, how do we introduce photo-blinking? Their idea is to use a DNA technology named DNA paint. DNA paint is a super resolution microscopy that uses a transient binding of DNA, which creates a apparent photo-blinking signals, which is shown in the schematic in the middle. And this leads to nanometer resolution, as shown at right. So the idea is each different types of amino acid will be labeled with a different sequence of DNA. And when imager strand labeled with different colors for different complementary sequence interacts with this so-called docking strand on a peptide or protein, we'll observe different colors appearing from the same protein molecule, but shifted by a few nanometers from each other. And I believe this kind of linear barcoding can be effective for single molecule identification, and looking forward to seeing publication from Peng-In and Ming-Jae Da. This DNA paint-based approach has inspired my group to develop another method of our own. Here we have a protein that has multiple residues for imaging, and say cysteine residues. The question we had was, could we determine the position of these residues using FRET? And the concern was, current single molecule FRET analysis allow us to deal with only one or maybe two FRET pairs, but not more. When there are multiple dyes present all together, it is not possible to determine the distance accurately. So we asked, could there be a new way to determine the distance from a certain reference point to here, R, one at a time, and then obtain Fred fingerprint like shown at right side? The answer comes from DNA paint. By attaching DNA docking strand to cysteine residues or any other residues of interest, we can make only one single Fred pair formed at a time and measure the distance in high accuracy for that particular pair. And this cycle repeats spontaneously due to the dynamic nature of a DNA hybridization. To demonstrate this idea, we have made a DNA construct. Protein work from my group is coming up soon. This surface immobilized DNA has two binding sites for labeled DNA. And depending on which imager strand associates, we get a different Fred state, as is shown in this time trace. When the two imager strands are separated by 30 nucleotide, it is actually very easy to distinguish the two peaks supporting our idea. But when the distance between the dyes become 20 or 50 nucleotide, the two peaks start to merge, and we cannot any longer differentiate them. That is, we cannot achieve a high resolution for structural analysis or protein protein sequence prediction using this scheme. So we had to rethink about this approach and came up with an updated method named the high resolution Fred via DNA exchange, in short FredX, where we have cysteine residues labeled with the docking strand in all different sequences. Well, we do not have a control on which docking strand will bind to which cysteine residues, but it doesn't really matter. What matters is when we introduce an imager strand of only one sequence, it is likely to interact with only one docking strand on a protein. And that isolation will allow for highly accurate determination of Fred and distance. And then we just repeat this several times for different imager strands after flushing the previous one. To test this idea, we made a DNA construct similar to previous, but this has two different sequences for DNA binding. At the end of measurement, using two different imager strands, we get a Fred histogram which shows a similar overlap to our previous. But because we did the experiment using one imager strand after another, we can separate into two distributions. From measurement using a first strand, we get a single peak as expected. And this distribution is broad, mainly due to chat noise, and we cannot do much about it. But since we know that this is from a single population, we can fit it using a single Gaussian peak and determine the center accurately than the standard deviation sigma. But essentially, we are determining the center with the accuracy of standard error, which is only 1% in this case. And it is a tenfold improvement. And we repeat the same for imager strand 2 and obtain a similar high accuracy. Putting the two fits together, we get this very sharp Fred signature or Fred fingerprint. Since Fred is sensitive over a few nanometers, we expect that we can get a sub-nanometer resolution with this 1% accuracy, even when there are multiple positions to reserve in a single structure. And our bioinformatics analysis using a human proteome also suggests that this resolution is sufficient to distinguish most proteins. To further demonstrate the sub-nanometer resolution, we designed a set of imager strands which have labels one nucleotide shifted from each other. And when this set of imager strand was used, we got a broad distribution of peaks, which are overlapping with each other, as you see from the middle figure. But this was obtained from 9 different measurements. So we can determine the position of each peak very accurately, as is shown at right. And there, the order of 9 peaks followed exactly what was expected. We envisioned that we can use this high-resolution technique, FredX, for protein analysis, protein structure analysis. A protein of interest will be labeled with DNA oligos, and from each round of measurement, we'll obtain high-resolution distance information. For cysteine residues, we repeat this about 20 times, which is an average number of cysteine residues of cellular protein in human cells. And we move on to another residue until we have obtained a sufficient fingerprint. And then finally, the information is compared to a database consisting of predicted fingerprints. Now, the final scheme from single molecule fluorescence is to do the end recognition of a protein. Sequencing or fingerprinting the entire protein is difficult in any case. So the idea is, what about reading from one of the ends of a protein? After identifying what is in there, you clip off the monomer and move on to another one. This requires Edman degradation, which involves cleaving the N-terminal amino acid from a peptide while not disturbing the peptide bonds between other amino acids. It is a well-established classical technique, but no one could realize it at the single molecule or in a high-throughput manner. And the remarkable achievement of realizing this was reported in 2018 by the group of Edward Marcotte and Eric Anselm from UT Texas. They combined Edman chemistry, single molecule microscopy, and stable synthetic dye chemistry. And labeled peptide were immobilized on a glass surface using the C-terminal sand, and multiple rounds of Edman degradation were monitored through total internal reflection microscopy. And then the obtained sequence was mapped back to reference proteomics. What is remarkable is this could be done for millions of different peptide molecules in parallel. The single molecule Edman degradation is a versatile platform, which allows for different ways of doing single molecule protein sequencing. One of such is to do sequencing by using N-terminal probes. In nature, there are several proteins that recognize the N-terminus in a sequence-specific manner. When these proteins are properly engineered for specificity and are labeled in different colors, each fluorescent labeled so-called N-terminal amino acid binding probe will selectively identify and report on a specific amino acid at the N-terminal end. And after each of the imaging, the N-terminal end is cleaved for a new round, as illustrated here. And this idea has been proposed by several groups, including Dan Marino from NIST and Ed Boyden from MIT. The experimental proof is yet to be reported. This was an overview of single molecule fingerprinting, and you may wonder whether we can do protein sequencing, not fingerprinting. And I would say the answer is yes. A promising one is also nanopore technology. So what is nanopore technology? It involves a tiny pore, which is the size of a few or a couple of nanometers, or sometimes a sub-nanometer. When an electric field is applied, a current runs through it. And when a polymer, like a DNA, translocates, it lowers the amount of the current. It's illustrated here. If the translocation is well controlled, and if the instrument is sensitive enough, you can even detect the nucleotide and identify them. This is a working principle behind nanopore-based DNA sequencing, and the same can be applied to protein sequencing. Yet, there are several challenges to realize single molecule protein sequencing using nanopores. One of them is a difficulty to make a protein translocate in a linear and controlled manner. So a common method so far has been to detect folded proteins by holding the protein in a pore for a sufficient amount of time. And then the electric current contains information about size, shape, and protein dynamics, but not the sequence. So a better way for sequencing would be single file threading and direct reading of unfolded proteins, like shown here. But like I said, achieving this for a protein has been difficult since many proteins have a stable three-dimensional folded structure, and also because the amino acid backbone of protein does not have a uniform charge, in contrast to nucleic acid. This complicates the dynamics of translocation to the nanopore. Addressing these concerns, a scheme that my group, in collaboration with Kay Stecker and Giovanni Maglia, developed involves a dipole structure. This peptide has a positive tail at one end and a negative tail at the other end. By the way, I should mention that this is similar to what Tudor Lucian's group developed a few years ago. When this peptide is inserted into a nanopore, due to a tug of war, the positive part being pulled to the right and the negative part being pulled to the left, it gets momentarily stalled inside the nanopore, which gives sufficient time to probe the nature of the peptide, or at least a part of it located near the constriction site of a nanopore. And as you see from the time trace, its residual time can be several tens or hundreds of milliseconds, corresponding to many data points to analyze. This is more directly shown from these simulations. At left, you see this dipole construct is suspended at the constriction site. At right, a peptide which has only a positive tail translocates through. The simulation is for a few nanoseconds, so this event happens very quickly in real time. We thought this platform would be a perfect one to probe PTMs, post-translational modifications, for its high signal-to-noise ratio. We chose the most abundantly found one, phosphorylation, to study. And we also chose the O-linked glycosylation, which is difficult to analyze using mass spectrometry. And this is the data we obtained using peptides with and without a glycan. When there's no modification, the level of current blockade is about 0.49. And a glycan is present, the current level goes up to 0.52. This increased blockade must be due to a larger size of a glycan. And overall, it indicates that we can detect the presence of a glycan using this dipole construct. When we repeated this for phosphorylation, we observed a similar current change. Phosphate is a very small chemical, but it has a charge which must be significantly influencing the current. And then after this observation, we looked for a condition where we can distinguish all three, non-modified, glycosylated, and phosphorylated. And we could indeed find such a condition by optimizing the salt concentration. And we observed three different levels in a time trace, and also in a scattered plot. And I should say this was the first time when the glycan phosphate could be distinguished using nanopores. This high sensitivity is very exciting and can be used for sequencing, I believe. But the design I showed, and also what others have tried, are mostly for peptide analysis. So you may ask whether we can do protein peptide sequencing using a nanopore. But as I said earlier, it's not straightforward to extend the platform for peptide to a protein analysis, which is a long polymer. An interesting scheme potentially solving this problem was proposed earlier by Jeff Nivala and Mark Eckerson, where they have used ClipXP to pull a protein substrate. Here, ClipXP is in solution, and it binds a substrate in a nanopore and pulls it down. Since the pulling is ATP dependent, the movement is rather well defined. But the process of ClipXP binding is stochastic. The amount of electric current running through nanopore and also ClipXP is unpredictable. So I think this scheme can be used for sequencing if the design is revised for better control, for example, by tailoring ClipXP to a nanopore. And I'm aware of a few research groups doing this, so I'm very much looking forward to seeing the progress in the near future. We are near the end of my talk. I hope this gave you a good overview of this emerging field of single molecule protein sequencing. And I can tell that when we can fully sequence, identify, and classify proteins with the ultimate sensitivity, we'll have a better understanding of the molecular biology of the cells. And this will open up an opportunity for single-cell proteomics and potentially allow for on-site, real-time clinical testing. And this tool should be cost-effective, easy to use, and it should come with the capability of fast readout. And it should be portable to make a breakthrough in clinical applications. I want to end by emphasizing that this research field is very young. This field needs new ideas from young scientists in particular. So for example, we had a first ever international conference on this topic only in 2017. With around 60 people joining who came to Delft from all over the world. We had a very exciting open discussion. It was so successful, so in 2019, we continued this series in Israel. In 2021, we will have the third one in Texas, Austin. So please stay tuned. Finally, I would like to acknowledge my lab members and collaborators for making this research possible. I'll be happy to answer your questions that you might have.
Thank you for your presentation, Chirlmin. It is now time for the Q&A. To ask the speaker a question, just type it in where it says type your question here and then press submit. So our first question, and this one asks, can the technology be used to detect all 20 amino acids?
Yeah, so this is a good question. So if we want to do de novo sequencing, we will need to detect all 20 different amino acids. But this will be possible only when we can translocate proteins in a very well-controlled manner. And currently, it's still challenging to do so. So we will still have to rely on protein fingerprinting, where we use some of the labels. So regardless whether you use a nanopore or fluorescence, we will have to do a labeling. And then the question will be how many different orthogonal chemistry we have for labeling. And so there are, it's known that there are 12 distinct side chain types, ranging from highly reactive amino acids to some of the ones that are very challenging to react. So highly reactive ones like lysine-resistant residues, they are easy to target, but there are other ones that with amide and alkynes, they are very difficult to label. All this together, right now, we have a method to label up to 6 amino acids, 6 different amino acids, which I'm aware that Eric Anselm's group in University of Texas Austin expanding to a larger number, but in the end, it'll be up to 12 different types of amino acids we can distinguish. So the answer is, if we go for fingerprinting, which rely on labeling, we'll be able to distinguish 12. But if we want to do de novo sequencing, then we will have to use a nanopore technique, which should come with the capability of very carefully translocating protein into a nanopore.
Excellent. Thanks, Chirlmin. Our next question asks, what is the accuracy of the protein sequencing?
So as for accuracy, it depends which platform we use. So taking example of Edman degradation, the accuracy is determined by 2 factors. One is labeling efficiency, the other one is the cleavage efficiency. And roughly to say, there's about 90% of accuracy you can get per amino acid. And then the question is, with this percentage, how much of a prediction power you have? And as I've explained earlier, we will need to do, we do not need the entire sequence of a protein. As long as we have some partial information protein, that will be sufficient to tell what the identity of a protein is. So there has to be computational work to improve the accuracy. So that said, the accuracy has to be as high as possible, but with the computational work, we will still able to identify protein.
Excellent. Our next question asks, what is the longest peptide which can be read?
This also depends on which method we use. Taking example of Edman degradation one more time, there in each round you have about 90% of efficiency of a cleavage, and so initially it's good, but then after say 10 rounds of a cleavage, you will have about 50% of a population that is asynchronized. That limits the Edman degradation based method up to 10 amino acid or so. But we can think of other techniques as such as nanopores, and if we could translocate a protein in a very well-controlled manner, then there's no limit in how long protein you can read. You can simply read from one end of one end of a protein to the other end. So there'll be no limit on this.
Excellent. Thanks, Chirlmin. Our next question asks, can the technology identify post-translational modifications such as glycosylation, for example?
Yeah, definitely. So this is what I have briefly mentioned in the end of my presentation. So using nanopores, which biggest advantage is that you can detect chemical tags, chemicals without modifications, and their glycan was big enough that we can detect it. But then question would be, what about other techniques? We can think of fluorescence technique. Do we have a chemistry for it? The answer is yes. There is a chemistry that allows specifically targeting glycan or phosphate as well. And the most classic one is beta elimination followed by microaddition. So there's a technique for it. But the challenge there would be that all these techniques are optimized for mass spectrometry, where the efficiency of conjugation isn't necessarily need to be close to 100%. Yet, for single molecule analysis, we should achieve as high as efficiency as possible. So we will still have to find a way of optimizing all these techniques, or probably we'll have to bring a entirely new chemistry for this.
Excellent, thank you. Our next question asks, how does this work with complex samples containing lots of proteins?
That is some challenge that people still have to find out. because the complexity of a protein is enormous with a dynamic range of 10 to the 8th or even 10 to the 9th. And so the initial work, I believe that we will have to focus on a simplified samples, such as very well-known biomarker, and then there we try to understand what type of proteoforms this might have. And then we move on to more complex one, but still the cellular proteomics analysis will be challenging. So we could obtain samples from more simplified ones, such as bodily fluid, where the dynamic range of a protein will be smaller. As for the actual proteomics, we will have to borrow some ideas from mass spectrometry, where they enrich proteins of interest and they deplete some of proteins that are not of interest, such as housekeeping genes. They are dominant in proteome, and they will waste all the data analysis time. So all these ones have to be discarded. But yet, all this description I'm making is a bit hypothetical because we don't have the working principle yet. But what I can imagine is that all the knowledge that has been built in from mass spectrometry is what we can really use for delivering this technique for actual use.
Excellent, thank you. Our next question asks, how does TOW ensure that the protein is pulled through the nanopore in the at the same pace?
Can you repeat the first part of the question?
So how does the TOW or tau ensure that the protein is pulled through the nanopore at the same pace?
TOW, tug of war. I didn't know what TOW was. The tug of war is a way of making the peptide or protein stored in the nanopore, but there is another driving force, which is electroosmotic flow, which is moving protein to the one of the side. So while tug of the war will give us some time to probe it, but in the end there's another flow that is making this protein translocating in a unidirectional manner.
Excellent, thank you. Our next question is in regards to the protein concentration from single cells. Do you think the nanopore technology can be used for detection of low concentrated proteins extracted from single cells?
This is a very important one because even if you have a highly sensitive technique, if the complexity of a sample is so high, then you may not be able to detect it. So if we have, for example, if you have a single nanopore and if you put it in a whole complex of whole proteins from a single cell, it's very likely that most of the time this nanopore detects a housekeeping genes. And so there will be two ways to do it. One is making arrays of nanopores, which Oxford Nanopore technology already have demonstrated. Another is, as I mentioned, depleting proteins which are not necessarily interesting is another way to do it. Another way of doing it, detecting low abundance proteins.
Excellent. Thanks, Chirlmin. Just a quick reminder to our live audience, you still have time to ask a question. To do so, all you need to do is type it in where it says type your question here and then press submit. Our next question, Chirlmin, asks, in the labelling of amino acids, how much would the PTM of amino acids affect the sequencing?
So this PTM, just to remind you, this post-translational modification will be sitting on amino acid side chain. And so it will interfere with the labeling. So for example, if cysteine or lysine residues are labeled, conjugated with the phosphate or glycan, then they are not going to be labeled properly. So it will appear as a deletion, which we'll have to deal with. But on the other hand, this gives an opportunity to detect PTMs. So after we labeling, amino acid of interest, such as lysine, cysteine, methionine, tyrosine, etc., we can carry out orthogonal chemistry that targets PTMs. Then we'll be able to read them as well. So it can be deletion we detect, which we deal with computationally, or it can be extra information we can use to identify what the protein is.
Excellent. Thanks, Chirlmin. Our next question asks, can you provide an example of a disease where specifying the protein composition could actually make a difference?
There are numerous protein diseases, including neurodegenerative diseases. Well, just taking a few examples of Alzheimer's, Parkinson's disease, etc. And as of Alzheimer's disease, the diagnosis of early onset of it can be done by detection of biomarkers such as tau proteins. And there you can see how single molecule protein sequencing technique can be used. Another example is Parkinson's disease, where alpha-synuclein protein or other biomarkers is known to have a central role in development of this disease. And the working principle there is the aggregation of this type of protein is leading to the disease. For understanding this and detection of this, we can use single molecule protein sequencing technique, because this aggregation is often caused by mutations, and also a change in PTM profiles, and that all is something we can detect using single molecule fluorescence. Single molecule protein sequencing technique.
Excellent. And sort of following on from that, Chirlmin, do you think any of these technologies will be implemented for medical applications?
That is something that I am asked quite often. There's a huge interest from clinical chemists and medical doctors. Whenever I present this work, they show high interest. This is something that I see it actually being done, but some people would consider this is too futuristic. But well, today is 28th of May 2020, when all of us are suffering from the epidemics, where I think making this statement about medical application very real. As you know, that PCR is the main tool for the test, which is very accurate, but this PCR has to be done by a trained scientist. And so this whole step is very slow, and antibody test has been shown to be attractive for the speed, but as we hear from the news, its reliability and sensitivity is very questionable. And so if we had had a single molecule protein sequencer already running, we would have been able to do quick and cheap tests for coronavirus, and then we would be able to get the result as simple as a pregnancy test does. And I think this could have made a very difference in this epidemics. So that could be one example where the medical application can be. That could be one example as for the medical application.
Excellent. Thank you, Chirlmin. I think you sort of touched upon this in a previous answer to a question, but this one asks, can we determine the amino acid modified by a particular post-translational modification using this method? Yeah, so this was partly answered earlier. From mass spectrometry, we know that some of the chemical conjugation methods, such as beta elimination and MAC addition, we can label phosphate very selectively and detect. And that can be, once labeled, we will be able to use a single mark fluorescence or nanopore technique to accurately determine where it is located. Excellent. Thank you. Our next question asks, do you think solid-state nanopores are promising for protein sequencing?
I think definitely it is a very promising one. The biggest advantage of solid-state nanopore is that we have a good control on this, and also it can resist to many of the harsh chemical conditions. Unlike DNA, we have to stretch proteins in a linear manner for protein sequencing, especially for protein de novo sequencing. And some of the biological nanopore may not able to tolerate the denaturation to be used. So I think the promising nature the promising aspect of solid-state nanopore is that it's tolerable on this harsh conditions.
Excellent. Thanks, Chirlmin. Our next question starts off with a little bit of wanting to get a little bit of clarification. So they ask if they've got this right in their mind, the Edmund degradation sequencing you described in your presentation looks very much like a DNA next generation sequencing, but for proteins. If that is the case, what is the sort of error rate for sequencing, and can you process different proteins at once?
So the platform I have mentioned, which is from Edward Marcos group, is indeed similar to next generation sequencing in that it uses surface immobilization method. The difference would be that in case of DNA sequencing, especially from Illumina, there you can do several rounds of sequencing. But in this case of the Edmund degradation, you can carry out only once because it involves degradation. And the currently known, the error rate is mostly from the degradation efficiency, about 90%, and that limits that leads to the limit of reading up to say like 10 amino acids. But I'm aware that Edward Marcos group and also Eric Anselan's group is improving this. So once it reaches to say 99%, I expect that you'll be able to read it further. And can we another question asked was that can we process different proteins at once? And this is indeed true. They have all different proteins or different peptides on a surface, and they are all imaged at the same time using wide field microscopy. So this is possible, and they from their presentation and necessarily in their paper, they have shown that they can process like 1 million different proteins.
Excellent. Thanks, Chirlmin. Our next question asks, can protein fingerprinting techniques like FRET be used to detect conformational changes in the protein due to binding, phosphorylation, etc.?
Yep. So as for FRET-X, I have presented as a tool to do sequencing, but it can be also used for structural analysis. So supposedly we have labeled the DNA to the surface of protein, and this has not perturbed a protein function and also structure. Then definitely we'll be able to track the protein conformation change when protein interacts with a substrate such as DNA or another protein. Also when the protein is modified with a phosphate or glycans. But then the big question would be that how severe the perturbation by DNA tagging would be, and that's something we still have to find out.
Excellent. Thanks, Chirlmin. Our next question asks, how will you discriminate single amino acids with nanopores? Biological nanopores usually read 4 to 6 nucleotide nanotypes at the same time, so resolution is still hard for nanopore protein sequencing. How do you plan to sort of solve this?
This is a very important question, so I have not elaborated this in my presentation, but indeed, just like DNA nanopore sequencing, protein nanopore sequencing, we also have to deal with several amino acids, roughly 5 amino acids. So then the challenge is we already have a complexity from 20 different amino acids, but then we have 5 different amino acid, 5 different amino acids all influencing signal at the same time. In the end, I think the answer is the computation. If by using machine learning algorithm, we will have to train the algorithm as much as possible. And another aspect is, unlike DNA sequencing, where you have to do de novo sequencing for in any case, we are dealing with we are dealing with a defined number of proteins. So when you are given a sample from someone, then as long as you know which organism it is from, then it is limited down to several thousand or tens of thousands different proteins. So that said, if you are interested in particular organisms like a human, then you might just train the algorithm by using some of the peptide sequence that you are expecting. So it's not like you have to train for the 20 to the 5th power of different sequences, but rather you may need to train the algorithm using just a few of the motifs that you are expecting from the proteins from an organism.
Excellent. Thanks, Chirlmin. Our next question asks, can you use this technique for in vivo cell studies?
I cannot see how we can do in vivo yet, but I am aware that people are trying to do in situ cell study on this. Let me just think, because this is not a common question I receive. So for example, when we have a protein inside a cell, and then if we can label them with the different fluorophores, we can introduce ClipXP machinery to read them. Or we could also have a proteins labeled with DNA, then using FIDX, we were able to identify their sequence. We could also think of using Edman degradation method for in situ. So there is several possible way of doing it, even though I do not see how we can use a nanopore method to do it.
Excellent. Thank you, Chirlmin. Our next question asks, when do you envisage this technology can be used as a reliable sequencing platform for cellular proteomes?
This is another question I get quite often, and the cellular proteome analysis will be the ultimate aim. I think that is going to take substantial time of, say, 10 years. But if we rather focus on more some other easier examples, such as targeting some of the biomarkers or analyzing proteome of a biofluid, then I think we can get there within about 5 years of time. But also it depends on how much of the effort we can put in together, which also depends on the funding resources. But I think some of the techniques are already very promising. So if everything works out, then in 5 years, we might be able to bring it into commercialization.
Excellent. Thank you, Chirlmin. Well, I'm afraid that is all we have time for today. I would now like to thank Dr. Chirlmin Ju for his presentation and for answering your questions. I would also like to thank the webcast sponsors, GenScript, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll join us again soon.